because I wanted to get back to you. We were talking about antigens a few minutes ago and what makes a good one with Robert and Peter. But Dr. Kaufmann, as I understand it, it's less of a discussion when it comes to virotherapeutics. So try to do a good job of differentiating mRNA vaccines and virotherapeutic approaches for us.
Yeah, let me try and pull those two topics together. Obviously, I will not repeat all the fantastic attributes of antigens that can induce anti-tumor immune responses, hopefully as specifically as possible. I would offer that one measure of neoantigen quality is derived from homology of these mutated tumor sequences to epitopes that we have identified from pathogens. The term is called molecular mimicry, but it isn't necessarily that an antiviral or antibacterial immune response then also finds cancer, but there is something about the antigenicity of viral antigens that we can use to predict the quality of a neoantigen. So that gives us a segue into the infectious disease world here and what we can learn from that. And while the field obviously has made significant improvements in how we predict which antigens that are mutated or which sequences that are mutated in a particular cancer should be included in a vaccine, I would pose that those methods still are not perfect at this point in time. So we have been thinking about modalities where we don't need to know which of these mutations are actually antigenic based on sequencing, which is taken at a certain moment in time. And that is why we're banking on the effect of viruses that essentially induce an in situ vaccination effect, where through a mechanism called epitope spreading, you essentially recruit the right type of immune system to the cancer, but then let immunogenic cell death and other immunological mechanisms do its trick. So the tumor essentially teaches us which are the right antigens. We don't need to understand it as long as they're effective at the end. And that allows us to generate, obviously, off-the-shelf products that have, faster availability and cost of goods advantages over modalities where you require initially taking of a biopsy and sequencing and a customized manufacturing of a product, even though obviously there are also improvements as we do that at scale.
Yeah. And that's a good segue to a question I had. I'm going to read to you. So there's a question that's come in from Christopher from our audience that I think is corollary or at least tightly tied to a question that I had for you guys, and that is how do we operationalize personal cancer vaccines, or how do we rationalize personal versus off the shelf, Joe, per the point that you just made. And Chris's question is, what do we see as the main challenges in reaching an automated quality by exception approach for personalized medicine so that produced doses can be released nearly as quickly as they are produced? So I guess let's try to jam those two questions in together and respond to that. Joe, we'll again, a big challenge, but Joe, I'll start with you since you're on my screen.
Yeah, well, I will let the others pitch why a personalized manufacturing approach is feasible, and obviously we have seen that. It is actually fascinating to see how BioNTech and others have been able to pull off trials ahead of recruitment timelines in the midst of a pandemic where that company themselves were slightly distracted. So obviously kudos to the field and advances on that. But we still believe that a off-the-shelf, low cost of goods kind of drug that is available immediately without personalization can offer advantages, in particular as we're thinking about, equity and inclusion and accessibility. This can be an advantage because it therefore enables us to make these drugs available outside of larger academic centers that might still be required at this point in time to really pull off effective personalized medicine.
Robert, you want to jump in next on that one?
Yeah, I mean, absolutely. I mean, a few years ago, you couldn't have used this approach. You couldn't have gone and synthesized a protein, or grown it in a cell culture, reactor fast enough to get to a patient, especially with, patients with the more aggressive cancers like the late stage ovarians, lung cancer, glioblastomas, or, pancreatic cancer. It just couldn't be done, right? You were talking on, multi-months to get to something that would be a personalized product. The advantage that we have now is that, we can, there are companies out there, there are things that you can do in-house where, you can turn around a sequence in, pull a biopsy and turn around a sequence in less than 2 weeks. With the average probably being more like a month now, right? So, but there are some really fast sequencing technologies out there. And the advantage that the mRNA space gives us, whether it's, linear RNA like we're using or circular RNA like, Peter's company is using, is that, we can synthesize those and put those into an LNP very, very quickly. So, we're talking now on the order of turning around a personalized vaccine in, a month to 2 months, well within the treatment envelope of even an aggressive cancer where that couldn't have been done in the past with anything that was a biologic or a protein-based product. Mm-hmm. But, I don't disagree with Johanna at all. I mean, having something off the shelf, and this is another place where precision medicine, is fabulous if you're talking about, an antigen, looking for a particular antigen, is that, Providence is not just looking at personalized vaccines, we're also looking at, common mutations where, we feel like, we're working on a whole suite of common mutations across ovarian cancer and glioblastoma with the idea that, we could have an off-the-shelf product that goes into a patient very rapidly, while the sequencing information and, the production of a personalized vaccine is going on. So, kind of at the same time, doing a sequenced approach, or maybe even doing a common antigen along with the personalized at the same time, as an approach, so that, you're getting kind of the best of both worlds there.
Yeah. Yeah.
And if I can break in there, at HDT we also believe in the other type of personalized, which is not necessarily the ultra personalized that we talked about here, but more of the personalized composition where characterization of the tumor will reveal which antigens are there, maybe common shared mutations, and the treating physician can then choose between a set of off-the-shelf cancer vaccines that are composed in a personalized manner. So that would be sort of analogous to the antibiotic resistance testing, where antibiotic is set in after lab tests. Everything is not the ultra personalized, which we're generally talking about when we're talking about personalized cancer vaccines.